Hi everyone, I'm Yihan Zhang, a postdoc in MoLab. My topic is impaired epithelial differentiation and host defense as a mechanism for pulmonary complication in drop syndrome patients. So first, I will introduce the background. Drop syndrome is an immunodeficiency disease characterized by recurring infection and high level of IgE. It is caused by stage 3 mutation in more than 2/3 of cases, and pulmonary sequelae are the main cause of death in drop syndrome. The previous study of drop syndrome were mainly focused on immune cells. However, bone marrow transplantation has been associated with mixed results in drop syndrome patients, indicating that, in addition to immune cells, the abnormalities of other cells in drop syndrome also need to be further explored. Therefore, this study explored the impact of stage 3 mutation on airway epithelial architecture and host defense function. So firstly, we derived airway basal cells from a drop syndrome patient and a drop syndrome mouse, which harbor different types of stage 3 mutation respectively. When exposed to differentiation condition, the proportion of sine cells decreased significantly in drop syndrome patient and drop syndrome mouse, while the proportion of secretory cells such as goblet cells or club cells increased. Moreover, bacterial infection significantly increased the phosphorylation of stage 3 in epithelial cells, and RNA sequencing represented a strong chemotactic and inflammatory response indicated by the expression of CXCL2, TNF-alpha, and IL-1 beta. However, epithelial derived from drop syndrome patient or drop syndrome mouse did not have strong immune response to invading bacteria. Importantly, immunodeficiency in drop syndrome patient and drop syndrome mouse made epithelial cell fail to initial neutrophil trans-epithelial migration in response to bacterial infection. This study will provide important experimental foundation for the development of novel therapy targeting stage 3 associated abnormalities in architecture and host defense in airway, mucosal, and lung. So finally, thank you for my supervisor, Hong Mei-Mo, and other collaborators. Thank you, Drop Research Foundation. Thank you.